Hi, I'm David Walsh in the laboratory of Stephen Hallam in the Department of Microbiology and Immunology at the University of British Columbia. Today we will show you a procedure for filtering large volumes of seawater for microbial biomass. We use this procedure in our laboratory to study microbial diversity in the marine environment. So let's get started.
To set up the filtration apparatus, use tweezers to place a grade GFD filter into the pre-filter housing unit. Loosely tighten the housing by hand. As a rule of thumb, do not over-tighten or the unit will leak once the pumping starts. Replace the carboy cap with the autoclaved cap and attach tubing by carefully inserting the internal tubing into the carboy without touching it. If necessary, use tweezers to guide the tubing into the carboy. Then tighten the cap. Pass the external tubing through the peristaltic pump. Attach pre-filter housing units to the end of both external tubes. Tighten the connection with pliers. To begin filtering the seawater, turn on the pump and set the flow speed to 1. Watch to ensure that water is being pumped in the proper direction. Allow 500 milliliters of water to flow freely through the pre-filter unit and into a 1-liter beaker. Turn off the pump and attach a Steravex filter to the male lure fitting at the free end of the pre-filter setup. Tighten the pre-filter housing snugly.